So Matt is amazing to go in the field with, as many of you were able to experience today on our morning mushroom foray. And it's great to welcome Matt back to Archbold, where he visited 3 years ago, he's been assisting with our fungarium ever since. So today he'll talk about some of his work with ectomycorrhizal fungi in Patagonia, and I'll turn it over to you, Matt.
Okay. Like this. Okay. Thank you, everybody. And thank you, Aaron, and for inviting me. It's a great pleasure to be here. So I'm just going to get started because I have a lot of things that I want to talk about. So I just wanted to start by saying that I love biological field stations because when I was an undergraduate, one of the things that first got me hooked in biology was working at a field station with the UC Bodega Marine Reserve in 1995, where I lived for 6 months. And then in 1999, 2000, for an entire year, I lived in Panama on Barro, Colorado island. And I felt like that's what got me really hooked on biology. So please keep doing what you guys are doing. It's awesome. I work now sometimes at the Ordway station when I can. It's a much better place to go than my office. Sometimes I have to stay there. So before I start, I want to say the work, of course, that I'm showing is the product of a lot of people helping out. And I want to highlight in particular my former grad student, Marcos Cayafa, two postdocs in the lab, Camille and Alia, Roseanne Healy, who's a mycologist who was in my lab for 8 years, and a former postdoc, Michelle Husino. They contributed, I would say, more than most other people for this work. And there are several others as well, but I just want to make sure I acknowledge them up front. So I tried to make this more of a natural history discussion, heavily focused on fungi. And first, I want to talk about sort of different hemispheres, some storytelling about biogeography of South America, because some of us are northern hemispherists and we don't know about it. And then also really a contextual part about why fungi are really understudied and why that impacts some of the things that we think about in studying fungi that might be different from some other organisms. Some of the things we have discovered in work that's been going on in my lab and with collaborators on the ectomycorrhizal fungi, South America, and then a story, sort of a segue to the end, talking about bird dispersal and sort of why that's interesting, or hopefully I think you will think it's interesting too. So first, a lot of this work is grounded talking about ectomycorrhizae. So ectomycorrhizae is this association between plants and fungi, and at the interface, they form this interaction at the root where the fungi, ensheathe the root, grow between the cells, and they share nutrients. Basically, the plants give carbohydrates. The fungi generally give nitrogen and phosphorus, although that's the simple version. There's probably a lot of other things going on. And so I'm going to focus mostly on those fungi. And although there's a lot more work going on in the quote unquote global South, but say most of the work that's been done on ectomycorrhizal fungi and plants has really been focused in the northern hemisphere, former Laurasia, and especially on pines, oaks, and Betulaceae, or Fagaceae, Betulaceae, and Pinaceae. Almost all the work up until maybe 10 years ago was really focused on those. It was kind of rare to have people having work on other systems. And of course, there's this whole other former supercontinent, Gondwana, and those things were all connected and they have a really different biogeographical history, which I'm sure some of you are somewhat familiar with. And in 2008, when I was a postdoc and had a lot more hair and no gray in my goatee, I went to Chile with my former advisor, who's shown here, Dr. Donald Feaster, and we went for a project that he had and collected fungi. And it was pretty interesting because even though the biogeography of the fungi is not generally well known, when you go and spend like 10 days collecting fungi, you see these things where you're like, this is totally different from anything in the northern hemisphere. So some famous things like Ceteria that are only known from Nothophagaceae in the southern hemisphere, but then things that are a little bit less obvious that those are southern hemisphere endemics. This one it belongs in its own family and it's known only from the southern hemisphere. And also this one is the same. And so Just more examples. You see tons and tons of this group, Cortinarius, many truffle-like fungi in Cortinarius. Alurina, which is more or less restricted to southern hemisphere. This one's not, but this particular species, this is a rare group in the northern hemisphere and is very abundant there. And then weird things like this sequestrate inocebi, which is sequestrate inosebis are only known primarily, well, there's one species from the northern hemisphere and all the rest are from the south. So certainly people have been studying fungi in southern South America before. So I don't want to make it seem like I'm the first one to ever do that. In fact, the first collections that made it into sort of Western lore all came actually from Darwin's trip when he was in the Beagle Channel, he was collecting fungi. So there's a long history of these people collecting, but I would say we've had some challenges in studying fungi. And sort of to step back, let's compare fungi writ large with plants and animals. I will make an argument, and hopefully you guys will end up agreeing with me that for plants and animals, I would argue that most of the lineages are described and have known distributions. There are exceptions to these things, but in general, that relationships among lineages are mostly well established with some exceptional things that stand out. In general, compared to fungi, you have excellent fossil records, and the morphological features tend to not be plastic over evolutionary time, comparatively to fungi. And when you do a side-by-side comparison with fungi, there are many lineages and species that remain undescribed. The relationships among the lineages, in many cases, even in the northern hemisphere ones, is not well understood. There's almost no fossil record, with a couple weird exceptions. And morphology, as we discussed this morning in the field, is just ridiculously plastic and changes. So that if you have just the morphology, you often can't even tell the order that you're looking at. So this has made studying fungi kind of challenging and continues to make it challenging. And of course, that translates into biogeography. So for animals, they make it into kids' books because we know what the main patterns are. So when my kids were little and we'd read books, try to sneak some biology in there, and you could find a book like this where it sort of tells you the main story of animal biogeography after this interchange between North and South America around 3 to 6 million years ago. The land bridge formed, and northern groups were moving south, and southern groups were moving north, and we see the evidence of that today. Okay, so that's the story for animals. I realize it's not that simple, but an oversimplified version. And for plants, I would say they're neglected a little bit more, but we know some examples of these very clear biogeographic patterns. So one that I'm going to talk about a lot is Nothofagaceae, which has this very strange seeming connection. Not strange if you consider the history of Gondwana, but where it's only in southern South America and Australasia. Similarly, you know the story of oaks very well. So oaks are, northern hemisphere, but here in the Americas, you do find them getting into South America, in Colombia. And this one, and I'm choosing these examples because they're all ectomycorrhizal plants that associate with fungi. Salix humboldtiana is in Mexico and has spread what apparently naturally to all of South America, or at least in some places in South America. So these are just examples. And for fungi, we really don't know that much. So this is a totally non-scientific way to do it, but I felt it illustrates the point. So this is the Great American Biotic Interchange, 1985. You can see plant things are mentioned a bunch, animal things are mentioned a bunch. And if you're a mycologist, you're so sad, right? Zeros, all zeros. Okay, I realize this is not very scientific, but you find this pattern over and over. By 2012, you get one time. We get spore one time and fungi one time. So we're improving over time, but still, comparatively, it's not looking like fungi have received very much treatment in this context. So this is the biogeography and ecology in South America, and we get one fungus, one spore. That's just unsatisfying. So, but yet at the same time, based on plants and animals and fossil record, we know a lot about what's happened. This is a really cool diagram from this paper from 2004 showing sort of the breakup of Gondwana over time. So this is millions of years going back. And I'm going to show you some some of these figures that are going to highlight southern South America and northern South America. And so what you can see is that northern South America was connected to Antarctica and Australia, New Guinea, much more recently over here, whereas northern South America hasn't really been connected to the next closest landmass for quite a long time. And that pattern's important both for animals and plants and also certainly for fungi. And I hope to convince you of that. Now, when you look in the same paper and you look at the patterns they talk about for plants and animals, they're slightly different between the two. But one pattern really stands out, which is southern South America is always connected to Australasia. So this is the biogeographic patterns that they found over, this is sort of review where they put together lots of data from lots of sources. And they find this very strong southern Gondwana pattern. Northern South America for the animals is much more related to the whole Arctic zone, whereas here it's related to the whole Arctic, but also Africa. So there's some difference in the signal between the plants and the animals, but this pattern is very clear. And I would say for fungi, we don't really have a synopsis about this. And so this is intriguing that, since 2004, we've had a good overview. And now for fungi, we don't. So in that context, it looks like the biota is really quite different in northern South America and southern South America, and also this pattern that southern South America is really connected to Australasia. Okay, the same thing seems to be true when you look at the ectomycorrhizal plants of southern South America. So I'm just showing some of them here, but this is really the big pattern. I told you oaks come into Colombia. Alders make it down the Cordillera of the Andes. And then there's a bunch of tropical plants that are ectomycorrhizal that are mostly concentrated in the northern part of South America. So Amazonia and north in the Guiana Shield. And then Nothofagaceae is down here like an island, kind of trapped by the Andes and being temperate while everything else is sort of tropical. There are some ectomycorrhizal host plants that are kind of dispersed across this, but they aren't big trees. They're like shrubby plants. Okay, so I was really interested in this system, especially after I went there, because I thought, well, this pattern really needs to be studied a lot more. And so the Nothofagaceae are 10 endemic species that are only in this region. They're the only native ectomycorrhizal trees that can host these fungi in this part of the world, except for the newly arrived Salix humboldtiana, which is very rare. It's not a big tree. It's only in riparian zones, so that doesn't seem to be super important in the system. And as you go closer to Tierra del Fuego, you get total dominance by Nothofagaceae. They're the only trees in the forest. If you go to Tierra del Fuego, you walk into a forest, the only trees are this one family, which is very unusual in the world, actually. And there's actually several endangered species, which I won't have time to talk about those really. So just to give you an idea of what these look like, very large trees dominating the forest and following this strong biogeographical signal. And so we went out and in the context of that, at the same time that I got interested in working in that system, I also worked on this review where we were interested in the biogeography of ectomycorrhizal fungi and what was known about them. So don't freak out because that's a big diagram that doesn't make a lot of sense. I'll explain it in just a second. And you can read about it in this paper, and there's some follow-ups that have dealt with this. But basically, what we did in this paper was we took all the different ectomycorrhizal lineages. These are fungi that separately evolved the ability to form this symbiosis with trees. And we looked at their biogeographic patterns by dividing up the world into these regions and saying, is that group of fungi known to be there or not? And so there's pretty simple, there's 4 patterns. The big red pattern, you can see it's basically all occupied. Those are the fungi that are everywhere. So they're simple. Like every place you go in the world, that fungal group is there. Then you go to these ones that are in orange, and you can see they're concentrated up here. Those ones are only basically known from the northern hemisphere. And then these ones have a very interesting pattern where they're in the north, they're in the far south, but they're not in the tropics. So that's the northern hemisphere or a temperate southern hemisphere pattern. And then this very small group over here, which is only in the southern hemisphere. And you can see, according to this, there's only 2. And so that's very curious because it kind of suggests that, maybe there's some questions that might arise from a pattern like that? So one is you get all of these missing groups. And so there's two possibilities. One is they could be actually missing from the tropics and the southern hemisphere. They could actually be missing from those places. Or maybe those places haven't been sampled very much, and a lot of these are little teeny truffles or cups down in the litter. And you don't really know which until you go and do some work to figure that out. The other thing is, are there really only two that are southern hemisphere endemics that kind of evolved this association in the southern hemisphere and they just haven't made it to the north? That's one, or are there more of them that are from there? And then the other possibility is maybe in those places there could be completely new lineages of fungi that haven't been found or documented before because those places are understudied. So those are some questions to keep in mind when we talk about this. And so in 2015, I took another trip, worked on several grant proposals, and eventually got some funding to work with collaborators in Argentina and Chile, and was able to go and visit Patagonia. So just to show you what some of these places look like, because many of us have not been there, it's a little bit hard to see these forests, but you can see there's different kinds of forests. So this is an evergreen forest composed primarily of evergreen Nothofageaceae. You find lots of deciduous or primarily deciduous forests of Nothofageaceae there. And you get these really pretty places. So if you're going to go on sabbatical, you should definitely go to a place like that if you can. So I've been very lucky. I've gotten to go multiple times. And just to give you a view of what this looks like. But as you get further north, you also get close to the Mediterranean climate. And so you get some really big differences over that large gradient that I talked about from Tierra del Fuego further north. And so I'm just going to give you a quick synopsis of some of the things that we found over many years of studying these forests. So we did some studies where we looked at ectomycorrhizal fungi directly on roots and also on soil. And then we also did a large number of fruiting body surveys over time. And there are two papers sort of focused on those two different things that were completed primarily by the postdoc Camille Trong, who I showed. And also using the data from several other studies from some of the collaborators of our group, there are some take-home messages that you can develop from looking at those studies. And one is whether you look at fruiting bodies or you look at the root tips, you find a very large number of species and high dominance by this group Cortinarius. It's exceptionally high compared to other places in the world. So you can see here, it's like, I don't know, 30% almost of the things that we did sequencing from fruiting bodies, 30% or 25% of the diversity here is Cortinarius. So you could go and walk out in a place like we did today, or even less, walk less than an hour. I found all these different Cortinarius in one site, like 20 meters by 20 meters, or maybe 30 meters by 30 meters. And so it's an amazing amount of diversity, and it's unusual in the world. And when you look below ground, you also see that these ones in red are all different Cortinarius species. So you can see that group is highly dominant in the system. You also see, because I love truffles, you find a lot of truffle-like Cortinarius, which is not something you find in the northern hemisphere. They're very rare. There's, a handful of species, and then here it's like 35 species or something are described. You also find a bunch of other ectomycorrhizal groups that some of which are northern hemisphere ones and some of which are not. So some P. physales, which are Ascomycota, many of them are cup fungi, some of them are truffle-like. You find a lot of different agarics, and this is one of the ones that's a southern hemisphere group only. So you don't find Descolia in the northern hemisphere. And so you can see these are the two lineages that showed up in that graph that I talked about before. One is the Austropaxillus lineage, and another is the Descolia lineage. And so those are very diverse in those systems and seem to have evolved this association in temperate Gondwana, because you also find them in like Australia, New Zealand as well. But we also found some evidence of some other interesting things going on. This is one that was highlighted in this paper in 2017, where we found actually two newly described species of this group Tromelodendropsis. There are basically 6 species, and 4 of those, based on sequence data, are only from southern South America. So that was a very interesting pattern that we did not expect. And Incidentally, we weren't sure what this was. In our previous review in 2010, we just had it as agaricomycetes, which is kind of like saying it's a bird. Um, we knew it was a bird or we knew it was a fungus, but we didn't really know. And so by finding these, we were able to try to figure out a little more closely what it was. So this group seems to have a lot of diversity there and almost no diversity in other places in the world. So it might be endemic to there. Another example is this genus Phaeohalocium. So these little teeny cups. So this is on a Nothofagus leaf right here to give you an idea of how small that is. So these are little teeny cups occurring on soil. And this phylogeny is just showing you that there's some from New Zealand. These red dots or red triangles are ectomycorrhizal root tip sequences from Australasia. And then the other four are 4 species from Southern South America. And we had enough material to name 2 of the species, and 2 still need to be named. But this is interesting because all the things that occur in this clade that are associated with ectomycorrhizal roots are only in the southern hemisphere. So this looks like another case of this association evolved in a different group. This ectomycorrhizal symbiosis evolved in the southern hemisphere and hasn't made it out of the southern hemisphere yet. Okay, and you find another pattern that stands out quite clearly is you find these things where that whole genus is only from southern South America and Australia, and not in the northern hemisphere. The whole thing, all of them are ectomycorrhizal in a group, but then you have 2 sister clades, one in the northern hemisphere and one in the southern hemisphere, and we find that a lot. So these are all examples that have that same pattern. And I just want to focus in on a couple of them. This one is a cool natural history little highlight because Roland Thaxter was a Harvard professor who visited southern South America. He had his sabbatical in 1905, 1906. He collected this weird gelatinous ball that has these very ornamented spores. He called it the white gelatinous ball in his notes. And when I went back to southern South America to the same sites with my advisor, part of it was to try to recollect this fungus because we only had 100-year-old specimens that were pickled, the Harvard herbarium. And we found it again. And we also found that it's very common. It forms these white mats all over the soil, and it makes asexual spores on those mats. But this little gelatinous ball is very hard to find. It's really, really tiny. So we were able to name it. And this genus was previously known only from Australia and in association with eucalypts. And so we now know that this genus is actually in 2 hemispheres, right? So that's pretty cool. And it's nice to go back to this natural history and read these old diaries and then find the fungus and give it a name. And we've continued doing some of this really basic taxonomy work because we're at this age of discovery is still happening for fungi. So this is another one where this group was previously only documented from Australasia based on 2 species that were only from Australia. And we've described a bunch of species and a new genus related to them. And they're all truffle-like. So that's part of why people don't find them. They're pretty rare and they're truffle-like. So it makes it hard to work on them. One thing that really was interesting to me is after many trips and doing a lot of sequencing of lots of collections, we also found evidence of northern hemisphere fungi that apparently reached southern South America through either animals or through a plant, some sort of plant bridge. These are 2, and we haven't really published on that, but these are 2 examples that were quite interesting. They have very close sequences to things in Mexico. And they're found all over, so they don't seem to be associated with introductions. They're not found near pines or anything like that. In this one, we actually have a collection from the 1906 records of Thaxter, suggesting that it was there in 1906 as well. But it's something these are only groups that are only in the northern hemisphere, except for 1 species that we found. So that's pretty interesting too, kind of an unexpected pattern. So sort of stepping back, I think we can say that we find the same divided sort of pattern that you find for plants and animals in the biota of these ectomycorrhizal fungi, and it follows those plant hosts. I didn't really tell you about it, but for my postdoc, I did some work in the Guiana Shield where we showed the dominance by these groups of fungi, and then ones that we find in the southern hemisphere are effectively all different groups. You find some species, like you find 1 or 2 species of Russula, but up here you find like 100. So you find really these strong patterns that emerge over time. And it's not very surprising, but clearly based on what I showed you in the books, it hasn't really been highlighted for fungi. So hopefully I convinced you that that's a distinct pattern that happens for the fungi in Southern South America. It's logical, but it hadn't been really shown or highlighted before. But another thing, after going out in the field with all these trips, is we found all these truffles. And I love truffles, so I'm like looking for them to begin with. But the diversity was very high. And that was interesting because, there's this really distinct morphology difference between things that are down here underground, where they have these morphologies that are totally enclosed, versus the ones that are their relatives that are these above ground things. And this is important from the perspective of dispersal. So these are the spores coming out. They're being released, right? You know what this, you guys probably have seen a spore print before, and that's a product of this ballistospory. But of course, if you're this one, you aren't doing any ballistospory at all, and you can't depend on airborne spores to get around. And so dispersal has really big consequences. Of course, I don't think I have to tell everybody this, but I will anyways. There have been a bunch of studies of dispersal in other organisms, but in fungi, I would say it's less known, but we do know that dispersal controls what those local communities look like from your regional species pool, just like it does in plants and animals. There's impacts on population dynamics, and certainly choose your favorite theory. And dispersal is going to be a really important aspect of what's happening in those theories. And we know that this has been shown also for ectomycorrhizal fungi, where if you look at the distance from the forest and the species richness here, you find these very strong patterns of dispersal limitation. And so dispersal controls what happens downstream. But then in Southern South America, we have things that look like this. So how are they getting around was one of the big questions that I had after going out in these forests. And so in the Northern Hemisphere, truffles tend to be pretty smelly. And you can take your truffle dog out and you're going to find them. And we know that a lot of different mammals eat them both in the Northern Hemisphere and also in Australasia. That's been shown. And of course, we know truffles are appealing to us as well for these odors. So this is a recent movie all about truffle hunters. They're mostly focusing on certain really high value ones, but certainly these odors trigger this response by animals. They dig them up and they eat them. Okay, well, that's all fine and good. And in the Northern Hemisphere, if you're someone who's interested in truffles, as we discussed earlier, you follow some squirrels around and you look where they dig, and then you look for the pieces that they missed, and then you'll get a good collection. If you're lucky and you have a truffle dog, you can bring your truffle dog out and they'll do all the work for you, and you just go right to the truffle. But things look really different in Patagonia. And after spending several field seasons there, I started seeing these patterns that did not look like things I would expect in the Northern Hemisphere. So here's one. It looks like a chicken came along and scratched there. You're like, that's weird. There's no chickens in this forest. So why is that happening? And you see things like this, where you find missing pieces of truffles, but there's no gnaw marks like you find in the Northern Hemisphere, like from mammals. And you also see this weird thing where you see truffles that look like other things on the forest floor, which is not something you would expect. So here's two different truffles that are really common, Cystangium and Phalangia. But if you look more closely at that picture, you'll see those are actually interspersed with berries. So that's a white truffle with white berries. There's a purple truffle with purple berries. And those are things you find around on the ground when you're looking for truffles. You're like, this can't be just by chance. When you go and you find Rulandiella, you sometimes find it with snail eggs. And it's like, looks quite similar. Why does that look like that? Why am I finding these things by chance? Here's another case. This is Dreamus winteri, which is a pretty common tree in some of those habitats. It looks an awful lot like this Cystangium over here that's a little golden. They're actually the same species, and they show this color variation, which is also weird. And then, just sort of hammering this home, here's another Cystangium right next to a beetle larva. And you're talking about a very dark, dense forest. Those things start looking pretty similar when you're under the understory. Here's a Descolia brunea, one there and one there with a slug. So I'm trying to convince you that there might be some visual thing going on. And you also see more chicken looking damage. I say chicken because that's my experience of looking at things that are scratching around. We used to have chickens at the co-op where I used to live. So to me, that looks like a chicken was pecking at it. But these are not things you see in the Northern Hemisphere in forests typically. And so I was intrigued by that. And so these things brought up some hypotheses. And Marcos was in the lab and he's from Chile. So we started cooking up some ideas to try to figure out if really there were some birds that might be dispersing these fungi. So the main hypotheses I'm going to talk about are using DNA sequencing from fecal samples will detect the community of fungi. And then also we would suggest based on fluorescence microscopy, if we look, that these are actually viable spores. So those are the things we wanted to test those hypotheses. And for this, we focused on two very common ground foraging birds, the tucao and the wet-wet. So this one's really, really common. This one's a little less common where there's people because sometimes people eat it. It's a little bit larger, but they're both very common. And as you go further south, you see them very like everywhere in the forest. So just to give you an idea of the distribution, this is the Nothofagaceae forest, more or less. And you can see those are highly overlapping distributions. So many Nothofagaceae forests over a wide area have these two species. So if you're from the Northern Hemisphere, you haven't been to Patagonia or to Chile, you won't be aware, but this is a really common bird and it's very charismatic. So tucaos make loud noises. You find them on like restaurant signs, kids' books, T-shirts, greeting cards. I mean, this is a very iconic taxon, and it's very evident because they make very clear calls in the forest. So just to give you a feel for, hopefully it's going to work. Let's see if it works. Okay, so they're ground foraging. They kind of hop around. You will see them just sort of, from my perspective, indiscriminately foraging, although I'm sure it's not indiscriminate. And you can see that the wet is just flinging soil. I don't know if you guys can see that, but it looks like he's got truffle rakes for his feet. Let's see, did it work? Yeah, there you go. So you can see he's flinging litter. He's got big old truffle rake feet. It's just like, it's made for a truffler to study that. And so we looked into the literature and looked into diet contents that had been studied for the, the stomach contents for these birds. And what we found is that there were studies that had found invertebrates and they had found seeds. However, they used seedling to look through these samples, as you might expect, and they looked through a dissecting microscope. So what's the take home is that you're not going to find fungi spores if you're not looking for fungi spores, and they didn't find any. So we went as part of my sabbatical and during Marcos's PhD, and we went to a lot of places. So we went over a 700-kilometer span of Chile. We went into the Andes, and then we also were in coastal forests, so covering a very wide area. So that's kind of like South Central Chile for those who don't know Chile. Puerto Montt is a reasonably sized city there, and you can fly there. And so we did fecal collections, and I am not a bird biologist, so I had to get a lot of help for how one might go about this. So we basically started off trying to capture the birds. And then what we realized is that was really hard. It was really hard to get large numbers of poops by capturing birds. So we actually watched the birds and we did a lot of passive collecting from the ground. So they have these very specific territories. You go there, you watch, and then you go behind them and there's poop all over. So you can, and I'm not a bird biologist, but I know what bird poop looks like. So I could collect it. And then what we did is for those ones that were passively collected, we would collect the poop, and then we would collect the soil directly below that, so as be as sort of a control. And then we could sequence from the CETA1 to be able to verify the ID of the bird. And so here's some of the early ones where Marcos was actually capturing the bird. We had to get an IACUC protocol, which I'd never want to do again. And we found a lot of poop. That's really the story. We found a lot of poop over a very wide area. And then what we did was, I'm not going to talk too much about this, glad to discuss it later, but basically we did metagenomics using fungal specific markers so that we could get a lot of information about which fungi were in the poop. And then at the same time, Marcos did a lot of microscopy on some representative poops, not every poop. And we suspended them in water, sieved them, centrifuge them, and then visualized them. And then he did some epifluorescence with a particular stain, which I'll show you in a little bit, to see if they were likely to be intact and alive. Okay, so the take-home message, what are the things that we learned from this? So we found that the fungal communities in the fecal pellets and the soils were quite different from each other. And so this is basically the take-home message. Poop and soil are different, which is probably not very surprising. There were lots and lots of fungi in the poop, and there's even more fungi in the soil. And the other thing that I think is important, even though we found this pattern we wanted to look at first to sort of convince ourselves that, yeah, we could look at these and it wasn't just what the soil. But we also paired these so that anytime we found a fungus in the poop that was also present in the soil, just to be ultra conservative, we discarded that one. So we excluded anything in those pairs where it was also found in the soil to make sure that we weren't overinflating the numbers of fungi that we were finding. And then we also found this really interesting, when we look at these 100 fecal sequenced feces over this area, we found this really strong pattern of biography, which is kind of what you would expect if there was a bird out there randomly sampling fungi and eating them, that whatever's available in there is going to be influenced by the geography, because the forests change quite a lot from there to there. They're still dominated by Nothofagaceae in all those forests, but they change quite a bit. Okay, so geography matters, which wasn't very surprising, and you find that it influences which fungi are showing up in these fecal materials. And then the other thing we found, which the haters told us we wouldn't find, was that there's a lot of ectomycorrhizal fungi present in those fecal samples. So it's highly variable. And so these are the two different birds are color-coded there. And these are just different samples. So there's definitely some samples that don't have very much. But then there's others where almost all of the DNA reads, all the sequences are basically ectomycorrhizal fungi, which was very surprising to me. I thought there would be plenty that were like this. I was pretty surprised to find some that were almost everything in the fecal sample that we got back was an ectomycorrhizal fungus, which was pretty surprising. The other thing, what we found is, so these are all those different lineages that I talked about. These are the ones that are the most common ones. And what you can see is some lineages show up in a rather large proportion. So not surprisingly, Cortinarius showed up. And you can see there's lots of lineages, and some of them are, in 25% or 10% or 15%. So there are lots of them. And in greater than 80% of them, we had at least one ectomycorrhizal fungus present. And so that was sort of convincing to me that there's quite a lot of these fungi in these samples. And whether you look at the number of species, so the number of ectomycorrhizal OTUs, or sort of estimate for species, molecular estimate for species, or whether you look at the percentage of the sequences in these samples, you find some really clear patterns. And it's nice because they actually match what we found on root tips, and they also match what we found by looking at fruiting bodies in those same systems. So these birds are eating a lot of different Cortinarius species, and they're eating quite a lot of them. There are some other ones that show up over and over again as well. And then what we did was so because people were skeptical of this, one thing that we really wanted to do was show that this is not just by chance, right? Because these fungi, a lot of those fungi I talked about, as I told you, they're dispersing spores into the environment. So you could just have a poop and then have spores land there. But if you're a truffle, you're below ground, right? And so you don't really have an easy way for your spores to randomly end up in bird poop, except for if a bird eats you. And so these are all things that we had collected as truffles. So we could confirm their morphology, and we had DNA sequences from the truffle itself. And what you can see is these birds eat a lot of different species. Some they seem to really like, and they eat a lot. There's many that are shared by both of the birds, so eaten by both of the birds. And when you go into those poops where you get the sequences, you can see the spores of some of these very distinctive species as well. So you get a sequence, oh, this one was loaded with, Descolia inferna, and lo and behold, Descolia inferna spores are all over inside of that one. So these are actual pictures from poop samples. I didn't just take them from the fruiting body and stick them in there. Those are real. Those really came from samples. So we did this enough to convince ourselves that that was a real pattern. They were really eating these fungi. And luckily, we found one direct observation where we actually got to see a jucal eat a truffle. So this is one of the most common species that you find all across all the sites. And so this is one particular N of one for him eating that truffle, or him or her eating that truffle. The other thing that I thought was really interesting is our protocol was not set up to sample for arbuscular mycorrhizal fungi, which are a totally different group, and it's very challenging for some groups to within there to get amplification of sequences. But we actually found a bunch of those as well. About 40% of all of our samples had some arbuscular mycorrhizal fungi DNA in them as well. And another thing that's pretty interesting about that is that there are actually people don't think about this, even mycologists don't think about this, but some arbuscular mycorrhizal fungi will make truffle-like fruiting bodies. So these are just 4 different examples that I've collected there, and they seem to be quite common there compared to other places. So that was pretty interesting and kind of a side, I would say a side thing that we hadn't intended to see. Okay, but one of the main questions is, does this actually mean anything? And one of the main ways we could ask is, like, are those viable spores? So a lot of studies have sequenced things from poop, and a lot of studies have looked at microscopes to see if those spores are there, but showing that they're actually viable is kind of challenging. And so we did this spore staining with propidium iodide in collaboration with a professor at UF who works in soil and water science. And what you can see is out of the spores we were able to visualize, about 50% of those spores were apparently viable. So when I say apparently viable, I'm going to try to show you what that looks like. So this is a light microscopy image, and you can see their spores, right? And then you stain it, and you can see some of them fluoresce and some don't. So just to show you again, the ones that fluoresce, they're dead. The ones that don't fluoresce, that means the spore is sealed, so DNA couldn't be stained, and we infer that that means they're still closed up and the DNA is intact. And so hopefully you can see that okay in this room. So we found about 50% of them were viable, suggesting that this actually has an impact downstream. The other thing that was really cool about the collecting, not just the birds and then collecting the poop from that, but randomly collecting poop off the forest floor as a way to get a lot of samples, is that we actually found 3 additional species that we hadn't been expecting to sample. We actually found that they had ectomycorrhizal fungi as well. And these are kind of closely related to the birds that we were trying to study, but this bird is not at all related. From my minimal bird knowledge, is quite distantly related. And this one is very widespread in that environment as well. And we found multiple samples where we found fecal material from that bird also had ectomycorrhizal fungi, which was not part of the study, but it was a cool sideline, I guess. And the other thing, I think it's made me rethink how these fungi might be attracting animals or interacting with animals by working in this system that's quite different from the ones we have here in the Northern Hemisphere. So we had, before COVID really rained on our parade, we had a lot of ideas of how we would be able to test whether this was active foraging or not. And we were really interested in looking at sort of whether there is reflectance on some of these fungi that's different, that might attract birds. We didn't really end up working on that more, but it's interesting and intriguing. And the other thing that's interesting is it seems to be emerging that there's maybe this biogeographic signal that in former Gondwana, that maybe birds were more responsible for truffle dispersal. So there are 2 relatively recent studies that came out just showing that when you look at the fecal material from these birds, you see spores, sort of suggesting that this might be a broader biogeographical signal. So I will say, if you're interested in that, the paper is out for a while now in Current Biology, and Marcos that was part of his dissertation. So hopefully I have convinced you of some things about fungi, that you see this biotic divide between north and south that follows what you see in plants and animals. Cortinarius is a big part of the story there. It's really diverse and very dominant. And that Northern Hemisphere fungi are mostly absent from that system, which is pretty interesting, I think. And then from the perspective of the birds, hopefully I also convinced you that these birds are eating lots of fungi and that that's a really widespread common interaction. The fact that there's truffles in these fecal samples indicates that there's likely some sort of active foraging going on, and that that's hopefully, I've convinced you that that's viable. So those spores could go on to form mycorrhizae and have a life of their own. Yeah, and so clearly there's a lot more needed in this system. So if people are interested, they can talk to me. So I think I'm probably over time. I just want to say there were lots of people who contributed to this work, the various pieces of the work. And so many of them are from Southern South America and remain contributors, and a couple of them have visited my lab, so it remains an active collaboration. And then got money and help from a lot of various institutions, including NSF and National Geographic. And I guess the thing I want to leave you with is whether you should consider whether you're a Northern Hemispherist, because I didn't think that I was so biased, but the more time that I've spent in Southern South America, the more that it's made me consider like how I see the map might impact what I'm thinking about. So I'll leave it at that. And thank you. And take questions if you have any.
So I'll run this one around and let's go on more.
Thanks, Matt. That was a really cool talk.
Thank you.
So I have a question and a comment. The comment is that, yes, that tortoise thrush that you showed is probably 50, 60 million years separate from the Tapaculo, which is a subaustin passerine. But the question, and the comment also is that tortoise thrushes are like global. It's a global genus, and they're all on the ground digging insects.
Yeah.
But digging for what we thought were insects, but probably they were digging for
truffles
maybe truffles also. So that could be a really cool thing to study. But the other question is, do you think truffles are just, or these fungi are just fooling birds into eating them since they're just peck marks?
Yes.
Or are they eating them?
Well, so this is one of the things we really wanted to test with like choice tests and things. But very hard to do even when everything goes well. And when there's a pandemic, it really sucks for that. So we didn't really get to test that. My hypothesis, or I guess my hunch, based on watching those birds a lot, is that they're just eating all kinds of things. It's very dark. The forest floor there is very dark, especially the further south you get, in the fall, which is when a lot of fungi are out. It's like really dark by 5 p.m. So in the afternoon, you can't see that well. And you see all these little berries and the truffles, they're intermixed. So I think they probably just eat everything. I think the fungi could still be very nutritious for them, but I don't really know that. So or no cost, yeah. The fact that we found so many samples where we found them suggests that they're eating whatever's there. And the other thing I didn't really talk about, because we didn't have enough sampling to say with great sort of certainty, because the way we sampled, we were really looking for a pattern that we thought was there. So we went to places that had lots of Nothophagaceae, but in the beginning, we took some samples in places there's some places near the coast that are what we call Valdivian forests. So they're dominated kind of by Nothophagus in patches and then Myrtaceae that are not mycorrhizal in patches. And it's kind of intermixed. And the proportion of Nothophagaceae just varies kind of what appears to be randomly. I don't know. And so in some of those forests, you don't find nearly as many ectomycorrhizal fungi. And, I think some of those samples are these ones up here where they might be getting other fungi, but they're not getting ectomycorrhizal fungi because there's just not that many around. So I don't think they go to a forest patch because they want those truffles. I think it's like those truffles are everywhere and they're all over the place, foraging on everything all the time. So that's my hunch. Yeah.
Yeah, thank you very much for the talk.
Yeah.
I want to ask you about the analysis you did for distinguishing soil and
Sure.
bird poop. Yeah, that one. I'm guessing, is this like principal component analysis?
NMDS. Sorry, I didn't go into the details. So this is NMDS. But it is an ordination analysis. Yeah.
And like I was wondering, like what variables, like you studied into
So
like
we only looked at the community of fungi for this analysis. We didn't look at any other variables in there. So this is just basically community space. And interestingly, we really wanted to be sure of this pattern, so we did it two different ways. So when you have a sample, you can basically say, oh, this fungus is present or absent, or you can calculate the number of reads, so sort of abundance-based. We know that there's some issues with abundance-based metrics with this because all these had to be amplified by PCR, which has all these issues, and that's kind of a known thing. So we analyzed it both ways. We analyzed it with just abundance and by just presence absence, and you find this pattern very clearly no matter what way you analyze it. So basically it's just community space. Because they have very large number of species in there, you get this pretty clear separation for the fungal communities. And I would say the other thing is this is all fungi. So a lot of the fungi that are there are like yeast, ascomycetes that are just everywhere all the time, and you can't really say too much about them. Although certainly poop has, fecal material has some enrichment of some of those groups, regardless of which bird you're looking at, if that makes sense. No. So it's like you basically make a giant matrix of number of samples and then number of OTUs or sort of species, and then you essentially look at how similar those communities are, and then you plot it. That's effectively what you're doing with these analyses. So it doesn't include pH or any other variables from those sites, just the communities of fungi. Yeah.
So, Hillary, and then take online questions.
So I'm trying to remember. There's a fungus that is found on sort of megafauna dung. I think it's Sporormiella or something. And I know we've had people writing to us now, saying, to what extent is this found on cattle dung here in Florida?
It's a dung
fungus, yes. Okay. I think it's called.
But it's on herbivores.
Well, it was supposed to be, I think it's a bit of a myth, but it was supposed to be very prevalent with megafauna, and now seems to have gone through a collapse in the sediment. I was getting back to the sediment question.
Yeah.
So it sort of collapsed in the Midwest sediments in the 14,000 years ago, 13,000 years ago. And I wonder if you've ever done any work on that, because it's a sort of, to what extent are megafauna providing fungal substrates for this sort of indicator fungus?
So I'm not sure that I know exactly which fungus you mean, but I would be interested to hear which one you're talking about, so we could look it up.
I have to go and look it up. Yeah.
Yeah, but I would say so. Birds aside, many herbivores have a very specific communities associated with the dung. And there's often sort of this successional thing that happens in them. So you get some fungi that are only found on dung and they just never are found anywhere else. And for a lot of those, it seems to be that the spores are passing through the guts of the animals, and they're often associated with large herbivores, in part because it's a very large pocket where they can exploit that space. But there's some very famous ones like Psilocybe cubensis. I mean, it grows on dung. You don't find it anywhere else. And that's one that's very famous because it's hallucinogenic and people are hopping the fence to get it. But there's other ones as well that are only found on dung. We don't seem to find that. These are very small fecal pellets, and we didn't follow them, so we can't really say anything about it, I guess.
Well, I just had the observation that it's always interesting to think about projects, and you gave us a wonderful example today of where it would be a sort of cross collaboration among the programs here, like birds or so. I was just really interested in what you're saying. I'll add one other thing. There is an extraordinary history of maps. We always assume that maps have the north set, and it's just maps from China were completely different. Maps from the early maps from the Middle East face south.
Right.
So we have this totally northern hemisphere version of the orientation of our map
Yeah,
projections.
this.
And it's always good to think about it. It's a very, the history of map projections is very interesting. Sorry, I just wanted to
And I
compliment.
mean, I guess I think about that. So when I went and told a bird, I did go visit with a person who studies birds, and I said, you know, I'm pretty sure I'm seeing this, and I showed that person the pictures, like, this can't be by chance. What do you think? They were just totally poo-pooed me. It was like, that is no way. They're just by chance, no way. And I was like, oh, now I'm getting money to study this because I'm going to show this person that I think they're wrong. So that made me inspired to work on it some more. But I do think we have these biases. And in the case of truffles, one of the biases is they smell strong and animals dig them up. And that's the story, you know, because that is what happens, I think, a lot in the Northern Hemisphere. But you know, there aren't a ton of ground dwelling mammals in those environments. And that also stands out. If you're in the forest enough, you just don't see a lot of mammal activity comparatively to here. So I do think it's good to think about our biases, and sometimes they can be really big ones, you know.
Right. So we'll conclude with one last question that was online asking if any of these truffles are edible.
Great question. I don't, so some of them might be, there are some that if I had to choose some to try, I know which ones I think I might be more likely to try because there's nothing poisonous in that group that's known. Some of them are in groups like Inospy where there's known to be highly toxic fungi in there, so I wouldn't want to be the test case. But basically nobody knows except for the birds. I mean, but I don't know. Sometimes birds can eat things we can't, so I guess I'd be very careful with eating them. Yeah, there are no native tuber species, which is the group that most people think of when they're thinking about edible truffles. As far as we know, there are no native tuber species in the Nothopagaceae forest.
All right. Well, we'll wrap up here. We have a reception outside. And if you have further questions for Matt, and let's give him another round of applause.
Thank you.